As Ryder mentioned, we've been collaborating. I think you were talking specifically about collecting hippocampal tissue from your patients for 2 years, but actually 4 years in total when you consider the whole scope of our collaboration using human surgical specimens. And this has been one of our greatest collaborations, and we're really happy to have Swedish colleagues as our partners. So I'm just the opening act today. I want to make sure that I move through so that we have time. Everyone can hear the portion that Kostas will present. I'm going to keep it at a relatively high level, but feel free to interrupt me if you'd like to ask any questions. And I want to tell you about, just very briefly, introduction into the work that's ongoing at the Allen Institute, kind of the overview of what our trajectory is, and a little context of why we're doing what we're doing. So at the Allen Institute, we have a program called the CellTypes program, which is by far the largest program in our portfolio. And this is a really foundational effort to understand the principal cell types of the mouse and human cortex. And the basis for this work is that after more than 100 years of trying to explore the cellular level morphologies of neurons, we still don't really know how many cell types there are, let alone what their functional roles are in the circuit. We want to understand how these cells contribute to the neocortical column and processing of information, and we want to know how much conservation there is between the mouse and the human to ultimately understand what makes us uniquely human. So this leads us to a dilemma, which is that if we ever want to understand comprehensively what is a cell type, we're going to be out of luck if we're only looking at a single dimension. And this is clear from, as I mentioned, 100 years of looking at morphology. That's not sufficient to classify and really understand discrete types. And so that's illustrated here with this cartoon about the blind people trying to describe what they see looking at an elephant. And of course, depending on which part of the elephant you're looking at, you think you see something different. And neurons are no different. They're very complex in their morphology, and they have very different features. There are many properties that define a neuron as a cell type: its morphology, its electrical properties, the gene expression of those cells, and the types of synaptic connections they form, among other things, but those being some of the most important in our mind. So how to really make progress and have a very comprehensive program that can really tackle this problem. Well, here's just sort of an overview of our overall objective. We have parallel programs to study both the mouse and the human brain, where we have built data pipelines to systematically, in a standardized way and in high throughput, explore these dimensions of neuron properties. So the electrophysiology, morphology, connectivity, and gene expression. And in the end, the output is we want to push this as a data product to the public, make it freely available to anybody with an internet connection to really accelerate progress in the neuroscience field. And of course, hopefully, some of these things down the road could have some sort of clinical application as well. And so we now have this Allen Cell Types database at this website here, where anybody can go right now and you can explore all of the diverse data that we've collected so far in both mouse and the human brain. In the human effort, this is only possible because of the great partners we've been able to build in the local Seattle community. So this started, as I mentioned, 4 years ago with this core group of neurosurgeons, including Ryder and Charles here. And I think that this has been extremely fruitful. When I started this job, it wasn't clear if this was going to end in 6 months and it wasn't going to be feasible, or if this would really be a long-term project. And I think, largely due to the great collaboration we've had, the excellent quality of tissue and the communication, we've been able to build this into a long-term program. Recently, we just added a bunch of new surgeons into this collaboration. This is not intended to be sort of pseudo-competitive at all. This is actually about bringing together diverse people with rare human surgical tissues to aggregate data in a way that we can solve a problem that is very hard to solve with any individual laboratory or source of tissue. And in fact, we've extended this even further to an international collaboration with other people around the world doing similar thing, and we're going to be hosting their data in a standardized format on our database. And that's something we're very excited about going forward that just started this past year. So how do we do this? There's some logistics to discuss about how to transport human tissue, prepare it so that we can study it in the laboratory. The main issue here is that it needs to be kept alive and in a pristine state where we can get cellular level access to living human neurons. And so here I'm showing the location of a surgical resection in middle temporal gyrus. These are the majority of the cases that we get, showing the screenshot of the location and the typical appearance of these tissues, often spanning multiple gyri. So we'll break those down into a smaller size suitable for mounting and slicing on a slicer machine that produces brain slices that are thin enough that we can examine on the microscope and do patch clamp recording. So to tell you just the basics about the patch clamp recording and the Institute infrastructure, we have a room that's very industrial-like. We have standardized equipment. We have about 20 electrophysiology rigs in this space. This is what the patch clamp rigs look like with holders for electrodes that we can then manipulate down onto the individual living human neurons. And this is basically our way of interrogating the properties of the cells. It's a two-way communication. We can design stimuli to introduce into the cell and then record back the response of the neuron. And by diversifying the types of inputs that we give to the cell, we can probe the defining electrical properties of those cells. And at the same time, we're introducing a dye to fill the cell and later recover their morphology and reconstruct them in great detail. So one of the very first things we noticed that was very striking in these studies was that the human tissue is incredibly viable. I think that not many people recognize this. This is maybe one hindrance to why not a lot of people do these types of studies. And in fact, that the human surgical tissues ex vivo survive about 10 times longer than rodent brain slices prepared under the same conditions. We don't understand the biological basis for this yet, but this is something incredibly encouraging, especially from the standpoint that these are rare tissues and that we can really do a lot with even a little amount of tissue. And so this figure here is just showing different time points where we do these measurements from these individual neurons. We inject current and see their firing patterns. The responses are relatively well maintained even out to 74 hours after the surgery has occurred. We never expected anything like that, but this led us to believe that we can build a robust data pipeline and really encouraged our institute leadership to invest in this and to go big and really build out this platform. So in a nutshell, we have brain slices from human patients. We're filling them, recording the activity of the neurons, reconstructing their morphology, and then also a part of the pipeline is, at the end, biophysical modeling based on the real electrical data, the real morphological data, to make in silico models of single neurons and also networks. And you'll hear more about that from Kostas. And then lastly, we do histology on every single case. Nobody is trying to make a claim that these are perfectly normal tissues. I think that's also a reservation. We know that this is not a normal human being. That's not going to be possible, but this is the best available material from the real thing, the real living human brain. And by doing this histological panel, we release that. We can do our own assessments and allow other researchers to make their own assessments about the integrity of these samples and what types of things might be altered. So we had our first data release after this buildup of 4 years of effort last October 25th, and now this is live. So anybody with an internet connection around the world can go there and access all of the data that we've generated so far. Apologize. There are ways to do feature search to try to look at the metadata, like if a patient, what is the age of the patient, what is the pathology type, the gender, all of these types of information we capture. You can filter the data based on that and do meta-analyses to understand how these things influence the properties of the cells. And here just showing sort of our little gallery view of every cell, and there's a lot more. This is just the first page, but showing sort of the electrical and morphological features that you can then click on and zoom in and interact with in a really nice way. This is just a gallery view of a lot of the different morphologies that we've captured out of these human ex vivo slices. Certainly kind of too small to really see in great detail, but just want to make this impression that there are many diverse types of neurons in the neocortex, and that overall there are a lot of types that are well conserved across species. So, we see similar types in rodent model, or also from the primate, non-human primate literature. And so that's reassuring that we can see these conserved types of cells, but also that there are types that we're seeing that are not described from rodent literature, which is really exciting. So to dive into this a little bit more, I just want to show you some more specific examples and tell you what we're doing with this data at a high level. We're trying to make connections to determine correspondence between the morphologies and the electrophysiological properties of cells. This is something that has not really systematically been done in human tissue. So for example, each of these are distinct types of neurons, the first being a pyramidal neuron in the neocortex, and then the second two being different types of inhibitory neurons in different layers. And their electrophysiological properties are, at least to my eye as an electrophysiologist, very distinctive, very different. So I could point out here that, for example, this middle cell, which is a basket cell, which would be typically a parvalbumin expressing molecular type, has fast spiking rate, very narrow spikes, as opposed to this pyramidal cell, which has a slower spiking rate and a reduction in the rate of spikes over the train. And then the third type, having this feature of after giving a downward deflection, then you see this in blue, this rebound spike. These are all very defining features electrophysiologically of these cells that allow us to take feature extraction and then cluster them and call them into discrete bins. We can now start to do comparative analyses across species. So for example, here, doing some comparative neuroanatomy of mouse versus human. And we can see a paired representation of either spiny pyramidal cells on the left or aspiny chandelier cells, which is one of the most distinctive morphological types in the neocortex, as well as a pair of Martinati cells. So just point out that human neurons, always shown on the left, are always much larger than the mouse neurons. That's not surprising. But in terms of their complexity, they're also far more complex. This is just the very first look, but allows us to start to get an inroads to what are the distinctive differences between species. Similarly, we can start to dive into the electrical properties of the cells and see differences between rodent and human neurons, in this case pyramidal neurons in the supragranular layers, which are the cells in layers 2 and 3 shown here. Obviously, there's a marked expansion of the human layer 2, 3 compared to a rodent. But the most important point is that some of these properties are completely crisscrossed. If we measure the properties of the neurons, like the membrane resistance of the cells, through the depth of the cortical layers, that these have very completely opposite trends in mouse and human. So I don't want to belabor the detail, but to point out that this could have very important implications for translational work. Certainly testing drugs on cells that have different properties is going to lead you to different conclusions about the efficacy of those drugs. And not just for excitatory neurons, but we also see clear differences in the properties of inhibitory neurons. Here just showing a simple example that this fast spiking interneuron type has a property of what we call sag, which is this lower deflection here in the hyperpolarizing current, which you almost never see in a mouse, which leads to things like a property called subthreshold resonance, which could suggest that these human interneurons are tuned to respond to different types of input or ranges of input compared to rodent neurons. How am I doing on time, Kostas? Where are we at? Oh, okay. And then kind of a last point is that this platform gives us the opportunity to target distinctive types that are not even present in the rodent brain. You would never, ever get this information from looking at this rodent model system. So, for example, von Economo neurons are a morphologically defined type of neuron that's been kind of famously described as originally like a primate-specific type. We now know it's not truly primate-specific. There are a lot of mammals with large brain size that have these cells in particular regions. In the human, you find them in the anterior cingulate or in the insula. We've been trying to target them in surgical tissues. It's incredibly hard to get exactly the right spot, but this is one example from one of the cases that we collected from Dr. Cobb's, where we were in a third region that has even fewer, but we think that we were able to target in the dorsolateral medial prefrontal cortex, perhaps the world's first recording from a von Economo neuron. It's extremely exciting for us. This would be really a big step forward if we could somehow gain a way to access these cells and really understand what they're functionally doing in the circuit. And then a final example is that in collaboration with other colleagues in Hungary who have been doing this work in human tissue even longer than we have, we believe we've discovered a novel brain cell type in layer 1 of the human neocortex that also does not exist in the rodent brain. And these have been named rosehip cells for their distinctive axonal morphology. So in these very high resolution images, there are little boutons, strings of boutons on the axon that are much larger and more distinctive than any other known type. They also have very distinctive electrical properties and gene expression properties shown here. So again, the collective data from different modalities is necessary to really come to this conclusion of what is a type and are these types novel in comparison to known types. And that's a very good first step towards really understanding these different cells and their functions. I've only spoken about a very small sliver of the program. There are a lot of things that we're trying, from electron microscopy to gene expression analysis to synaptic connectivity. So we're really trying to build this comprehensive program, and getting back to the point I mentioned at the beginning, that you need to have this multifaceted, multidimensional approach, or you really won't be able to understand the cell types. So I'd be happy to talk with you more. Anybody who wants to discuss this afterward, I can tell you more about some of these other programs, but I'll end it there for this first part to make way for Kostas. And just, I want to give a separate individual acknowledgement for all the people involved in building these pipelines. There's a lot of people involved, probably upwards of 100 people at the Allen Institute that contribute in some way. Especially thank the support of the neurosurgeons who helped to build this program, Ed Lean, who is the lead of the human cell types group, my team, and Christoph, our president and chief scientific officer, for really having the vision to go in this direction. Thank you. Kostas.